David is the Chief Scientific Officer at A*STAR, which is the Agency for Science, Technology and Research in Singapore, where he's been for a number of years. And previously he is also Scientific Director of the Ludwig Institute for Cancer Research. As I mentioned, he's the co-discoverer of P53, which is the gene that we are focusing around here. And previously he had done his undergraduate training in London at the University College London, and subsequent training in London, as well as Cold Spring Harbor laboratories in the U.S. He also is a professor of oncology at the University of Dundee in Scotland. David's received innumerable awards over the years, and just to highlight a couple of them, he's a Sir, which is like, as a Commonwealth guy, that's like cool. It's better than the Patriots quarterback. And in addition to that, received other awards, including the Paul Ehrlich Medal and a prestigious Fellow of the Royal Society in the UK. He's published more than 350 articles, and these have been cited more than 85,000 times, which is a spectacular testament to the importance of his work. And so it's with great privilege that I welcome David to give the opening plenary.
Well, wow is all I can say. I usually talk at rather dry scientific meetings, and this has a very different feel, but I'm very excited to be here. I want to thank David and his team enormously for setting up this meeting. I spent a day yesterday at Sick Kids, and I was amazingly impressed by the young staff that have been recruited here and the kind of excitement there is around the work that's going on. So I'm going to talk about our recent work on studying the level of expression of mutant P53. And this is very important, we believe, because we think that the mutant protein that's present and made by people who have Li-Fraumeni syndrome may be part of the process that's driving their cancer forward. So we want to understand the function of this mutant protein, how it's expressed and how it's regulated, as it's essentially a key target in our attempts to better diagnose and manage the disease and potentially seek its cure. So I'm going to talk about quite a lot of things today. First of all, we have very powerful models to study the Li-Fraumeni syndrome in both fish and mice, and I'm going to talk about those models and what they've told us about the regulation of P53 expression. I'm going to talk about something I think is important, that is that we've technically reached a level of sensitivity that we can detect the expression of the mutant protein in the heterozygous state. That is the state that most people with Li-Fraumeni are in, with one mutant copy of the gene and one wild-type copy of the gene. I'm going to talk about a new technology, which is to use an in situ approach to measuring RNA levels. And this, along with reporter mice, has helped us to understand a lot more about when P53 is expressed and what's controlling its expression. And I'll show that the expression of the proteins is in fact highly restricted. Most of the work in this field has been done in tissue culture, studying cells in a laboratory, but as we move out into real animals and in real people, we see that there's another level of control that we hadn't fully appreciated, and I think this is going to have a big impact in our work in the future. And then I'll talk about the effects of drugs and radiation on the expression of the mutant protein. This is obviously important. If we think the mutant protein can be a driver oncogene, then we need to be very careful about what kind of treatments we use that might increase or modify its function. And finally, I'll talk about a new tool that we've created, which are antibodies that can detect individual point mutant P53s, potentially giving us a new angle for therapy and diagnosis. So, as I think everybody in this audience knows, P53 is a very important stress-activated emergency brake for the control of cell proliferation, and it's regulated in a very complex way by many, many different stress signals. So in our normal cells of the body, the level of the protein is undetectable. In fact, if you study the proteome of a person, you won't find any P53. It's remarkable that this is such an important gene, the protein's expressed at such very low levels. But when you're exposed to UV light, for example, If you go out too much in the sun, or you're irradiated for therapy, or you're treated with a chemotherapeutic drug, or something goes wrong in the normal process of cell division, the p53 protein is activated in a very complex regulatory pathway that involves a negative regulator called MDM2, which is a ubiquitin E3 ligase. What those complicated words mean is the protein that switches off p53 and degrades it. And when p53 reaches high levels, it turns on a very large number of downstream genes. We christened it the guardian of the genome because that really is what it does. It responds to genomic damage and then activates all of these protective mechanisms. Some of those mechanisms act at the cell level to try and rescue the cell from the damage. Others act at the organismal level to, if the damage is too extreme, eliminate the damaged cell. And there's still a huge amount of study. We've been working on this protein for about 40 years now, and there's still an enormous amount we don't know. Why is that? It's because it's a very, very powerful molecule that does lots of different things. And when we think about the effect that happens in a family with a single base pair change in their whole genome, we realize how critical this protein is for cellular function. There's still a lot of debate about whether this model of p53 acting as a transcription factor, a master regulator, is the only thing it does. Does it do other things as well in the process of controlling and protecting the genome from damage? We do know, however, from DNA sequencing that one fact is certain for all time, and this is the gene that is most frequently mutated in all human tumors. So not only is it the gene that causes the Li-Fraumeni syndrome, but somatically, half of people with cancer alive today have a mutation in this gene. It's about 11 million people living with cells in their body that have this particular mutation driving their cancer. And it goes, as it does the inherited disease, across all tumor types. So we've come to realize, and this is the work of many scientists who are in the audience today, and it took time to realize this. Varda Rotter, who's sitting there, was one of the first people who really pushed this idea that the mutant protein wasn't simply a protein that had been damaged and lost its normal function, but that it had gained a function, that it was doing something else and could drive a cancer forward. And that data, those experiments have now been reproduced very elegantly in many different systems by many different scientists. And I think the whole community has finally come to accept that this idea that the mutant protein can act as an oncogene is really quite clear. And that has a very big impact on thinking about how we might treat disease which contains the mutant protein, and also how we might interpret different forms of Li-Fraumeni syndrome, because in some of the mutations in Li-Fraumeni stop the expression of the protein completely, and others create the expression of the mutant protein. And it's very important to try and understand whether that means that there are real differences in how those individuals will progress in their lives and what kind of treatment they might need. So, to me as a scientist, mutant p53 is a great target for us to develop therapy against. It's extremely challenging at the biochemical level. This is not an easy protein to target. It's not like an enzyme where we can make an inhibitor. But nevertheless, there are characteristics of it that make it extremely attractive. The first and perhaps most important, and what I'll talk about most today, is the accumulation of the protein in tumors. It's very specific. If a pathologist stains a section of human tissue, the level of p53 expression in the normal tissue will be very low. As I'll show, there is some, and it is detectable, but it's low, whereas in the tumor itself, it accumulates to a very high level. So if you imagine a toxic molecule that bound mutant p53, it would preferentially kill tumor cells. It's an oncogenic driver. It's one of the things that's pushing the cancer forward. We know that because its expression is retained in cancers. We don't lose mutant p53 during the development of the cancer. And we know a lot about it. Its structure and function and biological models are all very well advanced. And there's encouraging academic data, and as you'll hear during the course of this meeting, also some clinical data that suggests that this is a valid target for therapy. And there are many different approaches we can try and take to develop a drug that's going to recognize the expression of mutant p53 as essentially a differentiating factor between a normal cell and a tumor cell. So the first question is, where is mutant p53 expressed, and can we find that protein being expressed in the body? And this is a histology slide of a hair follicle from a mouse. And what we can see very dramatically is that in the mice that contain only the wild-type p53 gene, we can't detect the protein at all. I'll show later that the RNA for p53 is readily detected in this tissue. But if we look for the protein, we find that it's highly localized to a very small region of the hair follicle, just these few cells in brown here. The brown stain is an antibody to p53 that allows us to see the protein. So what this tells us in this very straightforward experiment is that the mutant protein is expressed at higher levels in these mice that contain 2 mutant alleles. They're homozygous, they have no wild-type p53, and we can see the staining, but it's very restricted. It's only in this small group of cells and not in all the other cells of the hair follicle. And the other type of approach I'm going to use, so you can see this beautiful staining. This is really a result of work in the lab where we've tried to optimize the condition for staining. You actually can count these individual nuclei of these cells that are positive for p53. And then the other technique that we and others have developed recently is to look at the RNA. So I guess most of you know that the gene is made of the DNA, it's copied into RNA, and the RNA is then translated into protein. And so what we're able to look at here is the RNA. And the RNA is differentially regulated to the protein. The characteristic of p53 as a protein is its very short half-life. But the RNA, of course, we can study as well. And so what you can see, again, looking at the hair follicle, that the RNA for p53 is also restricted. It's not present in most cells in the hair follicle, only in a small group of cells at the base of the hair follicle, which is we know where the dividing cells reside. And then the final tool I'm going to talk about is to see, can we get a grip on the individual mutant proteins? And so what this shows is a technique called Western blotting. And you're seeing here a cell line that has been transfected overexpressing different mutant p53s. And what we've made are antibodies that only recognize one of the mutations. So they're specific to the individual point mutation. This antibody recognizes the change R175H, one amino acid in p53. This recognizes another one, and this recognizes another one. So for the first time, this gives us a possibility to study the expression of individual mutant proteins in the context of the wild-type protein. So I'm going to talk mostly about what regulates, what controls what I've just shown you. How is it that we see these different levels of p53? And why do we see this incredibly strong staining in human tumors? So this is a human breast cancer, very intense staining with this anti-p53 antibody. The normal cells surrounding the tumor are not staining at all. And more than half of human tumors show this excess accumulation of p53. And often in surveys, it's high level of expression that's seen to be associated with poor prognosis. So high levels of mutant p53 in models promote metastasis and invasion. And the question is mutant p53 actually the most common driver oncogene in human cancer? So we know a lot about the mutational database, work from very many people, colleagues in the audience, and are curated very beautifully at IARC and other centers with a complete database of understanding of what types of mutation occur in human tumors. And these are from the somatic, that is the spontaneous mutations that arise in individual people's tumors. But the types of mutation that are found here are also the types of mutation that are found in Li-Fraumeni. And it's always been a feature of this system that we very commonly find missense mutations. That is, we continue to make the p53 protein, but with a single amino acid altered in its sequence. And the regulator of this pathway that we know most about are these proteins called MDM2 and MDM4. So normally p53 is being made as a protein, but continually degraded. And one of the things that's fascinating about this field is that the negative regulator is itself a target gene for p53. So this is like your air conditioning system in Singapore or your central heating system here in Toronto. It's a thermostat. And so we constantly regulate the level of this protein in an amazing way. We keep making it and we keep degrading it. It's kind of crazy. Why would you do that? But the answer is because you want to be ready. You want to be absolutely ready if something goes wrong. And if something goes wrong, you can quickly block this breakdown and rapidly activate this response. Similarly, when you want to switch it off, the system will accumulate the negative regulator, and you can switch off the system very quickly. And of course, the control of what's happening here are these p53 target genes that regulate the behavior of cells inducing senescence or apoptosis or differentiation, or helping in DNA repair. We know from genetic studies, and these are old experiments, and I should have revised this slide because there's some more elegant new studies, but nevertheless, these studies tell us the key finding, which is so important to understand Li-Fraumeni syndrome, and that is the difference between a mouse that has no p53 and one that's making a mutant protein. So both these mice, which in a way model Li-Fraumeni, they have a loss of p53, they both get tumours, lymphomas and sarcomas, at a much higher rate than wild-type mice. But when we have a mutant protein being expressed, we see this gain of function and the driving of these tumours into different types of carcinomas that are invasive and metastatic. So that's really the driving reason, this kind of experiment, why so many of us are interested in understanding better the role of mutant p53. Fascinatingly, when you look at tumour sections and samples, you start to find p53 mutation protein being expressed in all of us. So actually everybody in this room will have clones of mutant p53 in their skin. And this is just an example of such a clone in a fallopian tube of apparently rather morphologically normal-looking cells that contain this clone. This is not cancer, but this is a type of change that could increase the frequency of cancer. And you can see this in your skin. So you take a little biopsy of your skin, particularly if you've been exposed to sunlight, and you'll see these clones of cells containing mutant p53. Genetics suggests that the surrounding cells are very influential in controlling the size of these clones. So we tend to think about things in a cell, what we call a cell-autonomous way, p53 driving the cancer cell in a particular direction. But it's very important to realize that this process is also massively involved in interaction with the neighboring cells. And again, this creates one of the challenges in understanding Li-Fraumeni syndrome, because Li-Fraumeni people have a mutant allele present in all of their normal tissue, as well as a wild-type allele. And you can see that in some cases, it looks like you're looking at tumor history here. So this is a lesion from where you can see these normal-looking cells staining for p53, but then overt early serous ovarian cancer. And this is the cancer type in which p53 mutation seems to be absolute. In serous ovarian cancer, in the somatic population, in the normal population, you always see mutations in p53 in all of the cancers. So we've been looking at models, and one I'm going to talk about is a mouse model, and I'm going to talk about a fish model. So in the mouse model, we've used a particular p53 allele, which is to change a single amino acid, arginine to histidine at amino acid 172. And what I'll talk about a lot more during this lecture is the finding that we made to our surprise that in these mice we could see p53 accumulating in the proliferating cells in the small intestine in a very particular subcellular position. And that really has triggered all of these studies. But we also have looked at zebrafish, and you'll hear a couple of more talks today about zebrafish as a model. It's a very powerful model system, and we have zebrafish that essentially can mimic Li-Fraumeni. That is, they contain a point mutation in p53. And these mutant fish develop a particular type of tumor, Schwann cell tumor. They're prone to other tumor types if other oncogenes, such as the human mutant BRAF, is introduced into that background. And they have many advantages as a model organism, and human tumor cells can grow and indeed induce angiogenic responses in fish. So the p53 in the zebrafish is well described. In fact, p53 has been found more or less everywhere in animal species. We, some years ago, and others have found it in very, very primitive species. It's found in frogs and flies and worms and everything else. Very, very primitive things, something called Trichoplax adherens, which is basically a little blob, has it. And it's highly conserved. And in zebrafish, it has a normal p53 gene, it has a p63 and p73 gene as well. And interestingly, the point mutation that we've been working with is a methionine to lysine mutation at 214. This is a very typical inactivating mutation, a gain of function mutation in p53. And then interestingly, we found in zebrafish that they strongly produce an isoform of p53 that's driven by the wild-type protein, and that's just diagrammed here. But I'm going to use this system to study p53 expression, how the protein level is controlled of that mutant protein. So in order to do that, we set out to make a panel of monoclonal antibodies that recognize different positions on the zebrafish protein. To my annoyance, none of the hundreds of antibodies we've made against human p53 worked in zebrafish, and we're hitting the same problem, unfortunately, with the elephant, but it's okay, you can make new antibodies. So with these new antibodies, we were able to study p53 expression in fish, and the results were really very exciting. We could see these brown cells here. Every time you see brown cells, that means they've got p53. This is the antibody stain that we're using. And you could see that the zebrafish make p53 only in response to a drug. In this case, we're using roscovitin. In the untreated wild-type fish, we see no staining at all. This is what I told you about before. p53, the invisible protein, hardly detectable. And then if you want to check that your antibody is good, which the referees will always want you to do, you can use a morpholino, as it's called, an antisense to p53, and show that this staining here is completely obliterated by suppressing the expression of p53. So in this system, you can cut the fish many different ways. I'm sure good restaurants in Toronto. So you can see here in this type of section, we're looking at the whole body of this young embryonic fish, and you can see that the cell staining, the p53 positive cells, are highly restricted, and they're in fact restricted to regions of proliferation. So as I'll show later in the mouse model, there's this very tight link between making p53 and cells that are dividing. Of course, tumor cells are dividing, and all the cells we use in tissue culture are dividing. So most studies have been done, actually, of cells which are making p53 because they're dividing cells. But you can induce the p53 very nicely with drugs, or perhaps more relevantly with ionizing radiation, and you can see, again, this very restricted pattern of expression. You can combine these two agents and see a stronger induction of expression. But the critical experiment, so the conclusions from that is that the wild-type p53, as we see in human tissues, is induced by non-genotoxic drugs and by radiation. A sideline, it's induced also by drugs that block the p53-MDM2 interaction, and very importantly, the induction is cell-type specific. But the critical experiment, and the one that surprised me at the time, and Gigi Lozano has very similar results, was what happens when you have a fish that only makes mutant p53, can't make any wild-type p53, both of its genes are mutant. We expected, because mutant p53 was expressed at such high levels in tumours, that we would see the expression of the protein in the normal tissues of the animal, but we don't. We, in fact, see no detectable p53 in these zebrafish, but if we treat with a drug, they also induce very nicely in exactly the same cells as we see the induction of the wild-type protein. And you can see that here very clearly with ionizing radiation, where perhaps the signal is even stronger, and also with the combination. So what happens then in tumours? So the fish develop tumours, and you can stain the tumours with the antibodies, and what you see in these tumours that arise in the fish is very strong antibody staining. This is the N-terminal and C-terminal antibodies, and you can see it by huge accumulation of the protein by Western blotting in the tumour, and you can see by higher power staining the very high levels of p53 that accumulate in the tumour. So what's happening here is a tumour-specific stabilization of the mutant protein. The mutant protein is normally unstable in normal tissues and gets stabilized in the tumour. So in a way, we know something very important about cancer. Cancer contains a signal to p53 to stabilize the mutant protein. Now, to ask what's responsible for that degradation of the mutant protein, we can knock out target genes that we think might be important. And so in zebrafish, we knocked out both the MDM2 and MDM4 genes using zinc fingers. And you can do that and get viable animals because if you cross to a fish that's mutant, it can tolerate the loss of MDM2. The wild-type fish cannot. So if you knock out MDM2, the embryos don't develop properly, they die a p53-dependent death. So we know from these experiments, which have also been done very well in mice as well, that MDM2 is the critical negative regulator of p53, and without it, p53 will kill the animal and stop development. Now, the critical experiment is the one shown here. So now we have fish that have mutant p53 but lack MDM2, or we have the control fish that still have MDM2. So in the control fish, we see very undetectable levels of p53, but in the fish that have lost MDM2, the mutant protein now accumulates to high levels, but again, not in every cell, but in many of the cells. So the key regulator of the expression of mutant p53 is clearly MDM2. In this case, in MDM4, the other member of the family of the MDM2 E3 ligases doesn't seem to have any effect. So this is really critical. This helps us to understand what's going on. So we must imagine that in tumours, it's the negation of the ability of MDM2 to degrade p53 that's a critical step in the development of cancer. So we can conclude from these studies in zebrafish that mutant p53 is not stable in normal undamaged tissue. It's induced by DNA damage and drugs in the same way as the wild-type protein. The very critical point is that mutant p53 staining in tumours represents a tumour-specific activation of the p53 stability pathway. This is very significant, I think. This is a drug target. If we could get a drug, if you like, that was controlled in the same way as p53, it would be fantastic. If it was degraded only in normal cells and not in tumour cells. And then mutant p53 levels in the fish are controlled by MDM2, but not by MDM4. And something I won't have time to talk about today, but is very fascinating, is that when we treat the fish with radiation, the wild-type protein increases and then decreases very quickly. It's a pulse. But when we do the same thing in the mutant fish, we see this increase in level, but then it persists for much longer. And we suspect that there's some persistent damage that occurs in that background, which is something that we really are looking at in a lot more detail. So the question then, as we moved into mouse models, would we see the same things or different things? And when we started, essentially we saw the same thing. That is that there was a very high level of p53 expression, mutant p53 expression in the tumour. But when we look closely at the normal tissues in these mice with very sensitive antibody staining, we started to be able to, the first time, to detect mutant p53 in untreated animals. So there is a difference. It may be one of sensitivity of detection, but nevertheless it's very significant. And this has really opened a big door for us because it's allowed us to ask the question, where is p53 expressed in the normal body? And what you see is some residual expression and always associated with proliferation. So we've done this in a variety of different systems, and the pattern is so consistent. Here you can see every crypt of this small intestine has these positive cells at the base of the crypt. And we've gone through every tissue that we could get in the mouse and asked the question, where is p53 expressed? And so what you see is expression in the proliferating cells of the small intestine, the colon, the rectum, the thymus, the skin and hair. Everywhere there's cell division, there's p53. There's absolute link between the two. And that's, I think, very important in trying to understand the normal function of p53 and the regulation of p53, which includes the regulation of the mutant protein. Because remember, in all of these cases, I'm looking at the expression of mutant p53, not wild-type p53. Using this test, we could start to measure very precisely the level of p53, and this is highly relevant for Li-Fraumeni, because what we're able to see is that in this tissue that has we know can express high levels of the mutant protein, we can't detect any protein in the wild-type animals. So this is people with normal p53. But if you have just one copy of the mutant allele, we can start to see staining. So we went on to look at this in, again, many different tissues, and it's very consistent. So the level of p53 is highly dependent on the number of gene copies you have. And if you have one copy of the mutant protein and one copy of the wild-type protein, you can actually detect that, as is shown here. So now what we're doing is looking at all the different genotypes we can study. The knockout, we're very fortunate that this is a perfect control to show that our antibody is okay. There's no staining at all in the knockout. We introduce one wild-type allele, we can see no staining. If we have two wild-type alleles, a normal condition, maybe just a tiny, tiny hint. If we look at the two mutant alleles, very strong staining in these cells. Though remember that staining does still not extend up to the non-dividing cells. One allele over a knockout, again clear staining, and then what is the Li-Fraumeni level, the intermediate level where you've got one wild-type allele, one mutant allele, clearly less than in the knockout condition, but nevertheless clearly detectable. And this is very interesting. This tells us that the presence of the wild type gene is contributing to the turnover and half-life of P53, and that's why we're getting this intermediate level. And you can see this early in development. So this pattern of expression, of P53 occurs early in the development of the gut. And we've looked at this in many, many other tissues now. So we can see a bit more detail which cells are actually expressing, and we can go to higher power. And these very beautiful slides show that the type of cell that makes P53 is of two classes. There's what we call the basal stem cell. This is the cell that's going to give rise to all the other cells in the crypt. And then the cells that are in what we call the transit amplifying zone, these are the cells where the proliferation happens. Then these cells stop dividing, they differentiate and move up the crypt and differentiate into different cell types. So the work that we've done, and I don't again have time to show this, but we've shown very clear overlap of this type of cell with the LGR5, a very prominent stem cell marker. And this is true for all the tissues we've looked in. There's very, very consistent staining of P53 in stem cells and in transit amplifying cells. You can see the gaps here that aren't brown. These are Paneth cells, and these are cells that sort of act to support the stem cell, but aren't themselves dividing. And so we can extend this to other approaches, and we can do Western blotting to show that we can measure these different levels. We can show by immunofluorescence the same thing as I've shown by peroxidase. So here in red is P53. Here in green is a marker of dividing cells, a proliferating cell nuclear antigen, and you can see that they overlap very strongly. So again, it's these dividing cells that are actually synthesizing P53. And we can look at the effect of radiation on accumulation of mutant protein as well. So again, in the normal mice, we can't see any P53 at all. But when we irradiate, we do see P53, but it only occurs in those same cells as where we see the mutant protein. So this tells us that in response to radiation, P53 is still restricted to these proliferating, dividing cells. And the protein appears and then disappears over time following radiation. If you look at the mutant protein, you see that it also increases in level after radiation and then fades away more slowly. And you can actually see P53 working in these experiments as well. So this is again in vivo, and we're looking at P53 accumulation. We can see the high levels of P53 in the mutant, lower levels induced by radiation in the wild type. But when we look at the downstream gene P21, it's only induced by radiation in cells that contain wild type P53, and the cells that make it coincide with those that express P53. And this is work that we did in collaboration with Gigi Lozano, where we mimicked what had been done in the fish and showed in the P53 mutant mice, which were null for MDM2, we got really, really strong staining in these crypts. But again, the staining doesn't go all the way up the crypt, so it's restricted to this particular area. So in fact, you can measure very different levels of P53 in these mouse models. So you can see a very low level, intermediate level, higher level, a very high level, very, very high level. And only this, condition 5, mimics the kind of staining that you typically see in a tumor. So we really think that tumors are in this state. They've got a high level of expression of the mutant protein, and the MDM2 is not working to degrade it. So why is the P53 only expressed in these cells and not in these cells? What's controlling this? So we thought about a number of different possibilities. Maybe there's another E3 ligase that P53 is made but degraded in these cells. Maybe the messenger RNA is not translated, or could it be the control of the promoter? Is the P53 promoter being switched off as soon as cells stop dividing? And in order to do that, we were able to do two approaches. One is to use single molecule FISH and also qPCR, and we did this in the gut of the mice. We were able to separate the villi from the crypts, and I won't belabor it, but you can see very clearly here that the level of P53 mRNA in the crypt is high, but very low in the villus. The control here is the knockout mice. So it's extremely clear, and it's true for the wild type and for the mutant. So we know that the regulation of the promoter is the same for the wild type gene as it is for the mutant gene. The difference in expression is caused by the mutation in the protein itself. And this is really shown most dramatically, I think, by these beautiful RNA scope studies where we're actually able to stain for RNA as well as for protein. And what you can see, and again very nicely in the hair follicle, this very clear restriction of the p53 mRNA to the proliferating zone where the protein is. So it's not the E3 ligase idea, it's not the translational idea, it's the regulation of the promoter. And you can, so to take the approach another way, and actually something's been very interesting, before we did the RNA scope and got it working, I asked the question about the other E3 ligase, and I thought I was being clever, which I probably wasn't, but never mind. And we would use a proteasome inhibitor, bortezomib, to see what would happen. So remember, we can't see the wild-type p53 protein at all when we stain. It's undetectable. But what happens when we treat a mouse with the drug that's used to treat patients with multiple myeloma, a small molecule drug that stops the degradative machinery working in a generic way in mammalian cells? And what we see is really amazing. So suddenly, you can see p53 in these mice. So here's the control, and then we treat with bortezomib, and within 30 minutes of giving the drug to the mice, we can see the accumulation of the protein, but again, entirely restricted to the crypt region. So that means that if there was another E3 ligase, it would also be inhibited. We'd see further staining. So this is very clear. And actually, what we're measuring here is the pharmacokinetics of this drug. It comes in, it stabilizes p53, the drug's washed out, and the cells begin to degrade their p53 again. So this actually turns out to be a very, very powerful approach to studying drug kinetics for drugs that activate p53. And so we can compare all the MDM2 inhibitors that have been published and ask how persistent are they in activating the p53 response. But it also tells us something very important, which I think is going to be on my summary slide in a minute, and that is that though we see this beautiful, strong staining, it's very dynamic, because within an hour or two, it's gone away. So when you see those big, strong antibody staining cells, you think, oh, it must be there for a long time. No, not at all. In fact, it's within potentially a cell cycle that staining could occur. So if the cell cycle is 24 hours and you have staining for 3 hours, you're actually seeing potentially a cell in cycle. So we have to really reconsider carefully what we're looking at when we do these histological studies. And you can see, again, this differential between the expression of the mRNA and the expression of the protein. Because here we're looking at the wild type cells, untreated, no detectable p53, but plenty of messenger RNA. And here, when we treat with bortezomib, plenty of protein and plenty of RNA. And it's all in the zone of the cells where we see the expression of cyclin and Ki67 and PCNA. These are all other markers of proliferation. But they're not sensitive to the bortezomib treatment in the same way as p53 is. p53 is a remarkably short half-life protein in mammalian cells. The message is being used. The protein's being degraded. As soon as you block degradation, you can see it. As soon as you take the drug away, it will disappear again. This is the final one. I think, again, the same story, essentially, this ability to see the protein when you treat with bortezomib in the same place as where the RNA is. So here's the proliferating zone, Ki67. That's where the p53 mRNA is. You can't see the p53 protein in these wild-type animals, but as soon as you treat with bortezomib, you can see the protein appearing, and you can understand that this is what we've all known from tissue culture, that p53 is exquisitely regulated by protein turnover, and the messenger RNA is being made, but only in the cells where p53 is needed, the proliferating cells. So in conclusion, we think that p53 is highly regulated at the mRNA level. The p53 promoter has not been studied at the level it ought to be. It's clearly switched off extremely actively in differentiated cells. It's only present in dividing cells. So we think p53 is doing something very much linked to division. p53 staining can disappear in 3 hours, therefore very dynamic. Mutant p3 levels are tightly controlled at the level of gene dosage, mRNA levels, and MDM2 activity. And variation in mutant p53 levels may affect the severity of Li-Fraumeni syndrome. We just begin to think we're getting to the point where we could pluck a hair from the eyebrow and stain for p53 and determine the level of expression in an individual person. So the final thing I want to talk about, which I won't spend too long, because I'm sure you'll want some coffee, is just recent work generating new tools. I am a great believer, as you've probably gathered from this talk, that you have to innovate technologically all the time if you want to make progress. And as we heard in the very inspiring talks in the beginning, you should never give up. You have to be very persistent in science, and then you will find a new and amazing thing. So this is an example of such persistence, because I tried to make these antibodies about 20 years ago and spent a lot of money and failed completely. The challenge is to try and make an antibody that will recognize a single amino acid change in a protein. And I got inspired to do so again by conversations with my colleague in Singapore, Kanaga Sampathi, who said basically, oh, stop being pessimistic and let's have another go. So we started again, and we changed the approach. And the key turned out to be, in trying to make antibodies that recognize these single amino acid changes, was the design of how we would immunize the animal to encourage the animal to make the right sort of antibody. So typically what we've done in the past is take a small synthetic peptide like this, couple it to KLH, a big protein, and immunize with a complex. And we didn't get the right sort of antibodies. But a colleague in Singapore had been using a different approach, and that was what we call in-frame insertion, so into the thyrodoxin protein. So it's a bacterial protein. We can make it in very large amounts. Right in the middle of the protein, there's a loop that occurs naturally in the protein. And into that loop, we inserted p53. And then we inserted actually not one copy, but 3 copies of the piece of p53 that we wanted to make antibodies to. And we positioned the mutant amino acid in different positions in the peptide so that we had maximum chance, we felt, of trying to get an antibody that would recognize this particular change. And I was very excited right at the start, because even when we looked at the serum from the animals before we made the monoclonal antibodies, we could see that something interesting was happening. And anyway, I mean, to carry it a long story short, we've been totally successful in this approach, and we've got these amazing antibodies now that recognize individual mutations. So all of these dots are different mutants of p53, and the 2 that are positive here both contain the R175H. Here, 248Q, 273. So these are the antibodies we have. We've mapped precisely how the antibodies bind using PET scanning. And I just show this in higher power. It's very interesting. So this is the mutation that we're after, R175H. And if we change that H to alanine, all reactivity goes away. But the epitope is more complicated than that. It is looking at these other amino acids that are the same between the mutant and the wild type, but this change is sufficient to give the antibody its specificity. And that was really confirmed by using a technique called Phage Display, where we have a random library of all possible peptides and then fish them out with the antibody. And as you can see, all the sequences that we fished out contain this histidine rather than arginine, this critical amino acid that we're trying to distinguish from the arginine in the phage. And they all contain this lineup. You can see this H, C, P, H, H, and the clones all have H, PHH, or HPH, or HPHH, etc., etc. But they all contain, every single one of them contains histidine in the right position relative to these other amino acids that anchor where we are binding the antibody. So these work extremely well in Western blots. They work in immunoprecipitation. They work in immunofluorescence staining. So you can just take a cell line now and sort of genotype it for its p53 by staining with these antibodies. And then they can distinguish, even at a given position, amino acids, so that our antibody to R248Q doesn't recognize R248W. And most importantly, and this is the critical slide, we can actually do it on paraffin sections. So you can get an ordinary section now from a tumor, and hundreds of thousands of sections potentially, and stain them and say which particular mutation does that contain. Now, because of the nature of the mutational spectrum in p53 and the number of antibodies we'd have to make, this is a restricted approach, though I'm very inspired by this meeting, and maybe one should just go crazy. But anyway, I was thinking 10 antibodies would be good, because there's 10 mutations that are very frequent in p53. And you can see that you can detect that this section has actually got the 175 mutation. This section's expressing mutant p53, but it doesn't have the 175 mutation. And these antibodies are allowing us to do this. And I won't belabor it, but essentially we started by going through tumours where we knew the sequence and could confirm that the antibody would recognize only this mutation and not these other mutations. So these three all have 175H. These all have other mutations. They're expressing high levels of p53, but the antibody doesn't react with it. And then ultimately we went into completely blinded samples and were able to prove by sequencing that the antibody was picking up the mutation. And fortunately for us, from an experimental point of view, these epitopes are conserved between mouse and human because they're in a very conserved piece of p53. So the same antibody that's going to be very useful for human research can also be used to study in these mouse models. And again, you see the mutant antibody is staining the mutant protein exactly in the right place at the bottom of the crypts here, and also reacting with cells in tissue culture. So what's next? And this is my last slide before coffee, except I'll show a picture of the lab. So the first thought of this is to try and extend how many mutations can we look at? Are all amino acids possible? Or is it that some changes are too subtle and we won't be able to make an antibody to them? We don't know the answer to that. Can we study the structure of these? Can we crystallize these antibodies with these peptides and understand more? Yes. Can we use modern techniques of antibody evolution to make them better and stronger? Yes, we certainly can. Can we do more? Is it interesting to immunize with these? Could we see some effect of the antibody? Is there even a possibility of turning these molecules into something with therapeutic activity? And there are two amazing breakthroughs that are happening in science at the moment with antibody engineering. One is that a group of antibodies have been discovered that will go inside cells. Traditionally, we've always thought about antibodies as only being able to be used for surface antigens. So they're no good for p53 because it's in the nucleus of the cell. But recently, we and others have been working on antibodies that can go inside the cell. We don't know how. So this is really sort of exciting new stuff. And then the second thing we've been doing is to use our knowledge of E3 ligases like MDM2 to form linker molecules so that you can grab onto a protein, and with this linker molecule, it's got like two faces. One face binds your protein, the other binds an E3 ligase. You can make the protein be degraded, and this is called the PROTAC approach. So at the moment in the lab, we're trying to take these antibodies, convert them to the single-chain format, fuse them to the antibody that goes inside the cell, and add on the thing that will target them for the E3 ligase. And then, if I'm very lucky, we'll have a protein that when you put it on cells will go inside and degrade mutant p53 specifically. And I think that would be very exciting. And we'll stop there and just say a big thank you to everybody.
So we have maybe one question, David, that was a masterpiece, and then we'll break. And we'll also have, just before we come back, some explanation of the breakout sessions, which will be helpful. Anybody have? I knew that Dr. Privis would have a burning question. Go for it.
Carol.
How does viscobitine stimulate expression of p53?
I've written many papers on this, all of which are ignored. It's a CDK. it's a CDK7/9 inhibitor, and the p53 system is remarkable in its ability to transcribe itself and other genes in the presence of an almost complete shut-off of transcription. So when we were at Cyclocell, we published a lot of papers on this, and essentially you can get to the point where very tiny levels of RNA synthesis are happening, but it's all to do with p53. And I think this is an amazing sort of system to have a rescue. You know, the cell's in great distress, and it wants to make the right protein and do the right things. I looked at it quite hard. It looks to do with the phosphorylation of the C-terminal domain of RNA pol 2. And there's a bypass. p53 does not require phosphorylation of the CTD in order to have transcriptional elongation. And somebody should be working on this. Somebody very skilled in the art.
So we're Thanks very much, David.
Thank you.
That was-